So just welcome the next speaker, Dr. Belinda Phipson, currently senior postdoc in Professor Alicina Asnack's group at Peter MacCallum Cancer Center, focus on developing statistical methods for analyzing RNA-seq, single-cell RNA-seq, and DNA methylation data. She will talk about beyond single sample applications of bioinformation adventure. Just welcome.
Thank you. Thanks for the invitation to talk here today. I'll just share my screen.
Yeah.
Oops, no, I don't want to do that.
Yeah, it's okay. Yeah.
All good? All right, I'll
Good.
get started.
Yeah.
I just make that a bit smaller. All right. Right. Yes, so I'm really excited to talk in this forum today, and I'm going to tell you a little bit about what I've been working on over the last few years. And so when I first started working on single cell data analysis several years ago now, the main question that we were really interested in answering was, what is in my sample of interest? And with my collaborators, we were really interested in characterizing all the cell types of the mouse kidney. But it became quite clear to me that we're going to be using single cell RNA-seq to ask specific hypotheses. So we want to know what is different between my condition. So what is different between my disease group and my control group? And this is becoming a much more common application now. And although we still have to characterize what is in our samples, find out what cell types there are and so on, we can then move beyond that and ask at least two questions of the data, if not more. And one of them is which genes are differentially expressed within a cell type, but between my two conditions. And another question that we haven't really been able to answer before with bulk RNA sequencing data is which cell types are under or overrepresented in my disease group, for example. So this was work that I did in collaboration with Dr. Alex Coombs, and we wanted to provide a comprehensive roadmap of the cell types in the developing mouse kidney. And so we designed an experiment, and I convinced Alex to do biological replication, so have more than one sample. And we profiled kidney pairs from 3 genetically identical mice that were raised in the exact same environment. And this was quite a few years ago now, and these experiments were quite expensive at the time, and we had about 6,000 cells, and it was one of the larger data sets that I had worked on at that time. And we did a really careful analysis with the tools that we had available to really deeply characterize what we saw in these mouse kidneys. And while I was going through the clustering and all that kind of analysis, what I could clearly see was that although we had genetically identical mice, there were still differences in gene expression that we could see when we were clustering that were stronger than the cell type differences. And so we see that sample to sample variation in the gene expression signatures. And not just at the gene expression level, but we can also look at how variable the cell type proportions are. So these are 16 cell types that we characterized in our kidneys, and we've got the cell type proportions on the y-axis, and the points are just the cell type proportion estimates for each of my 3 mice. And you can see that for some samples, we've got some cell types, we've got quite variable estimates of these proportions. And I was really interested as well about how on earth we could analyze a data set from a designed experiment that had been profiled using single cell technology. And around this time, Evangeline Sim approached me to form a collaboration to understand the developing human heart. And we work with Evangeline and Enzo Perello to design this experiment where we had human heart biopsies from fetal samples, from children, from adults, and from a disease group. And we had at least 3 samples, so 3 different humans in each of these groups, and they will all be different in terms of genetics and environment. So quite a lot of variation, as you can imagine, is what we would see in this data. And if we just sum the cells to create pseudobulk samples and did a principal component analysis shown here in this little plot with PC1 versus PC2, we were quite excited firstly to see that the samples do kind of cluster together. So the fetal look really different to the other samples. And generally, we could see that there was quite a lot of variation in the data, but we can see those time points coming out. So I thought I'd briefly talk about the workflow that I did for clustering these really heterogeneous samples. And I'll start off with the counts matrix. And from there, what I did was I did QC on the cells for each individual replicate separately. And then I decided that I would analyze the time points individually. So I looked at the 3 biological replicates in the fetal group, for example, and performed gene filtering to get rid of those really lowly expressed noisy genes. We used SC transform normalization from the Syrah package, which I looked at a few different options, and that was definitely the one that performed the best in terms of clustering. Then we performed data integration across those 3 biological replicates, dimensionality reduction, data scaling, and so on, and then performed clustering with Syrah as well to determine our cell types. And then we needed to find genes that define those clusters. So looking at genes which are overexpressed in one cell type compared to all the others. And we did that with marker gene analysis. We looked at gene set testing, lots and lots of visualization with t-SNEs and clustering trees and violin plots, and looking at some of the markers that had been published in mouse heart as well. And I left it to Enzo and Evangeline to do the interpretation of the clusters. And so armed with all of that, we managed to define what the cluster identities could be in our 3 time points separately. And some of these cell types were really clear, but some of them were not, and there were little question marks next to the annotation. And so what I asked Enzo and Evangeline to do was to try to combine the cell types that were most similar so that we could define these broad cell types so that they were in common between fetal, young, and adult, because ultimately that's what we want to compare. We want to compare between fetal, young, and adults. We've got to have some similarities there. And so we had 7 broad cell types shared across these that we observed on all of our data sets. And we also observed a small population of erythroid cells, which I'll come back to later. So from here, this is where you can do the sort of more interesting analysis and find out more about what you're trying to test. And so we further refined the cell types that were present at different developmental time points. We wanted to know which genes were differentially expressed within specific cell types across the developmental trajectory, as well as sex differences, because we had male and female samples as well. And we also wanted to know whether the cell type composition changed. So I'll very briefly step through each of these questions, and I will spend a tiny bit more time on question 3 because that's sort of work that I've been looking at lately. So what I chose to do in order to refine our cell types within our broad clusters is that, so all these pinkish cells are cardiomyocyte cells. So I grabbed all the cardiomyocyte cells across all 9 samples that we had, and I basically reran my analysis workflow. So redid my cell filtering, and I was a bit more stringent this time. Gene filtering, normalization, integration across my 9 biological replicates, and then clustering at fairly low resolution to avoid overclustering. And so this is an example of what the reclustered cardiomyocytes look like in the 3 different developmental time points. And then we repeated this for every cell type. Next, we wanted to perform differential expression analysis. And when I was doing this analysis at the time, Helena Corral published a paper in Mark Robinson's lab, published a paper on bioRxiv, comparing different approaches and also advocating for a pseudobulk approach. So this is a pseudobulk view of the data. Basically, each of these points represent the summed cells within a cell type for a particular biological replicate. And the colors show the different cell types, and we're quite pleased to see that these do indeed sort of cluster together. And you can see that, for example, the fetal samples do look quite different to the adult and the young. And so we performed differential expression analysis and looked at the differentially expressed genes across the developmental trajectory. And we also were able to find genes that were differentially expressed between males and females within cell type and within groups. So for the vast majority of the cell types, there's not many genes that are differentially expressed between males and females, but we noticed that in cardiomyocytes, which is one of the key cell types in the heart, that there were quite a lot of genes that were differentially expressed between males and females. So that was quite exciting. So finally, I just wanted to talk a little bit about how we tested for differences in cell type composition. So you can often see plots like this in the literature where they've sort of reduced all the information to the three groups here that we're interested in. And if you did a classical test of differences in proportions, every single one of these cell types would be highly significant. And the reason is because we don't take into account sample to sample variability when we do these classic tests. And so if we expanded this bar plot out showing all the cell type proportions, what you'll notice is that the cell type estimates are highly variable within our groups. And so we need to develop a test that takes into account the fact that we have a large amount of variability around these cell type proportions estimates. So we wrote a function to do this, and we call it Propeller, and it's available in the Speckle R package, which is on our Oshlight GitHub. And basically what we have is, what we do is we calculate cell types proportions for every sample and every cell type. We perform a variance stabilizing transformation on the proportions matrix. And then we formulate a linear model. So we use linear modeling here with our variables of interest. So that would be, for example, our time point, fetal, young, adult. And we test each cell type individually. We do empirical Bayes t-test for 2 conditions and empirical Bayes ANOVA for more than 2 conditions. And we do a multiple testing adjustment to account for the fact that we are testing on lots of different cell types. And so this is the top most differentially abundant, or whatever we want to call it. It's the cell type that is most different between our groups, and it's the erythroid cells. And this is a really cool, true positive. Basically, the developing red blood cells have still got their nuclei, and because we have done a single nuclei protocol, we've managed to capture some of those in our fetal samples. But once red blood cells are mature, when we're older, they don't contain nuclei anymore, so we don't observe them in young and adult. And so I'll just quickly show you the other results. So the next most significant, it's sort of ordered from most significant to least significant. The next most significant was immune cells, which also makes perfect sense. The fetal samples haven't had a chance to develop their immune system, and as we age, we get exposed to more viruses and so on, and we develop a much more diverse repertoire of immune cells. And then for cardiomyocytes and fibroblasts, they weren't quite significant in FDR 0.05, but it looked like they were more overrepresented in fetal samples compared to the others, and relatively underrepresented in fibroblasts compared to young and adult samples. And the remaining samples the remaining cell types were not significantly different. And you can see why the estimates are just incredibly variable within our groups. So I think single cell RNA-seq for the design experiments is becoming very common now, but I still think analysis is really challenging. And I think it does depend on your tissue. If you have a very heterogeneous data set, it will be a bit more challenging. I think biological replication is important, and biological replication at the individual level, not necessarily at the individual cell level, particularly for things like differential expression analysis and cell type proportions differences, where we need biological replication to get those measures of variability around our point estimates of gene expression of cell type proportions. I think we need to be really cautious when interpreting cell type proportion differences, and particularly when we don't have biological replication, just because everything can look significant. And we have developed Propeller, which seems to work pretty well to do this, and it's quite robust and flexible. It will take you don't have to do any kind of pre-filtering of your cell types or anything. It will work when there's lots of zeros or when there's a cell proportion of 1. And we found some interesting changes as well in the developing heart. So I just wanted to give a shout out to all the amazing package developers, and these are just some of the packages that I use when I analyze my data. And without all their work, this would be so much harder. And finally, acknowledge people involved. This is our Zoom lab meeting. We haven't seen each other for about 8 months now. We're all working from home, and we're headed by Alicia Oshlag. And I also want to acknowledge my collaborators, and I'm funded by an NHMRC Emerging Leader Investigator grant. So thank you for listening, and I'm happy to take questions if we have time.
So let's have some questions. A fantastic talk. The first one, how do you conduct the test when a cell type is completely missing from the biological replicate under one condition, but abundant under another condition? Just fill it with zeros? Thanks. This is one proposed by one attendant.
Yes, so you can still, so the way that it works, so if we perform that variant stabilizing transformation, which is arcsine square root transformation. And so in that example of the erythroid cells where we had zeros in the young and the adult, what ends up happening is that we had non-zeros in the fetal group. And because we're doing empirical Bayes, using an empirical Bayes modeling framework, what we do is we actually use the information of the variances in the fetal group to assign a variance for that particular cell type. So we don't actually estimate variances within each group. It's like a pooled variance, and we can utilize the information that we have so for example, for the erythroid cells in that fetal group to get that measure of variability. And then we also do sharing across the cell types to further stabilize those variance estimates.
So, several questions in the chatting board. Someone said he was told that combining bulk RNA-seq and single RNA-seq together would get accurate cell proportion. For single-cell RNA-seq, would lose some cell, especially the large ones. Does this hold true now? Just one question. In the charting board, yeah.
I haven't actually looked at that myself. I guess you would want matched bulk RNA-seq and single cell data. We did a nuclei protocol, so that meant that we were able to get those bigger cell types. So cardiomyocytes are typically missing from single cell protocols. So at least we knew that we had captured those cardiomyocyte populations, but I haven't actually had a look at combining bulk RNA-seq with single cell RNA-seq, because we would have to do some kind of deconvolution of the bulk RNA-seq to get an estimate of the cell type proportions, unless you had, so I'm not sure. I haven't done that.
Okay, so I still have one question. Just how to accurately different differentiation just the difference of cellular status or the cell type differences. So for example, the same cell type may have different expression profile during the adult and just in embryo stages, right? Just for the same cell type. So how do you can accurately annotate the cell type and the cellular status? I think the two factors are confounding together.
So do you mean, so we did, well, the way that we annotated our cell types was we did the fetal separately, we did the young separately, and we did the adult all separately. And so because we also weren't sure what we were going to find in those tissues as well, we weren't entirely sure of all the cell types that we were going to capture. And there wasn't at that time, there wasn't really like a very definitive like list of genes that could identify every single cell type. So
Yeah,
I feel like for us, but I feel like even though the genes may be changing, I think that a cell type identity should still be pretty stable, I hope.
okay. You
Across
suggest in the cell type is a predominant effect, right? Yeah.
so
Okay. So it just has another question. It's about Jinyo. In the presence of patient variation, how do we combat that when we're doing differential express gene analysis? The
I think it's
patient
really
variation, yeah.
I think it's a really tricky problem actually, and I don't know if I would even recommend my approach that I took, because it took a long time to analyze the data in that way to try to reduce all that variation between our individuals. And I feel like, especially with the Human Cell Atlas efforts, if we could annotate our cells using reference databases, I think that would be maybe a better unbiased way to go about it. So we take each individual sample, annotate it, and then have a look to see what, you know, what we have across all of our patients or whatever. But it's really tricky. It's a really tricky problem. Yeah.
Okay, let's have a last one short question. In your talk, you just mentioned pseudo-bug-seq. Does it mean you calculate the average VAF in one sample? It's about pseudo-bug-seq.
Yes.
Yeah.
Yeah. So VAF is
Variation. It should be variant
that
allele
means
frequency. Yeah.
yeah. So I didn't do any genetic profiling
Okay.
or whatever it's
It should
called.
be RNA gene expression, right?
Yeah,
Not
it's RNA-seq,
variant.
it's gene
Yeah.
expression. So what happens is, so apparently, I mean, no, I shouldn't. So in the paper, in Helena Krau's paper, they look at averaging the gene expression, so on the log scale, or summing the gene expression counts. So I summed rather than taking the average, because when you sum counts, and if that counts are negative binomially distributed and you sum a negative binomial variable, you still get something that is negative binomial distributed. So I felt quite comfortable to take the sum and then perform the usual differential expression analysis that we would do with like tools like EdgeR or DESeq2 or Lemma, Voom plus Lemma. I think what I did was I summed, then I did a Voom transformation, and then I ran a Lemma analysis following that. So that's what I did, and it looked like it produced pretty good results.